In 1993, Kary Mullis walked onto the stage in Stockholm to receive the Nobel Prize in Chemistry, the highest honor science can bestow. But here's the thing. This laureate didn't believe HIV caused AIDS. He denied climate change. He claimed he'd been abducted by aliens. So how did a man who rejected scientific consensus invent the very technique that revolutionized all of biology? The answer forces us to reconsider what we think we know about scientific discovery. Because Mullis didn't succeed despite being a terrible scientist. He succeeded because of it. To understand this paradox, we need to go back to where it all started at Berkeley lab in the 1960s, where a biochemistry student was far more interested in LSD than lectures. Kary Mullis just wasn't cut out for traditional science at Berkeley. He skipped classes to take LSD. Lots of it. His PhD dissertation was so full of jokes, the committee refused to approve it until he cut all the wacko stuff out. But the psychedelics shaped how he thought. As he later said, it did trickle into his science Ache 22. He wrote a letter to Nature magazine chronicling the entire universe from beginning to end, and they published it. A joke letter from a grad student in one of the world's most prestigious journals. After graduating, Mullis just bummed around. He took a job in a cardiology lab but quit because he was horrified by how many rats they killed. He worked in a bakery. He wrote fiction. He had no interest in being a scientist. Then one day in 1971, a man named Tom White walked into that bakery. White worked at Cetus, a biotech startup, one of the first in the world. They got to talking, and White offered Mullis a job. His pitch to his bosses was simple. Hire this guy. He's an excellent synthetic chemist. I knew he was good because he'd been synthesizing hallucinogenic drugs at Berkeley. Cetus was trying to develop commercial DNA tests. It was an exciting time. Scientists were just learning to manipulate DNA with molecular scissors that could cut it at specific sequences. The vision was to create diagnostic tests that hospitals could run in hours instead of weeks. But Mullis wasn't on the cutting-edge stuff. His job was to synthesize short DNA fragments, the probes used in the tests. It was slow, repetitive, mind-numbing work. And maybe because he was bored, he became unbearable to work with. He picked fights with everyone: receptionists, security guards, his colleagues. He was a womanizer. While married, he once even threatened to bring a gun to work. Nobody wanted to be around him. Then, the company brought in a new machine. It could synthesize DNA fragments automatically, doing a month of his work in a single day. Suddenly, Mullis had free time and access to materials he could experiment with. And because nobody liked him, he started taking long weekends at his cabin in Mendocino County. On a Friday night in the spring of 1983, Mullis was driving up Highway 1 to that cabin. His mind wandered to the DNA testing problem back at Cetus. Their new test worked faster than the old methods, but the signal was just too faint to be reliable. Then a thought struck him. What if instead of inventing a better telescope to read a license plate from the moon, you just created more license plates? His mind raced. He could see DNA chains floating around him. Blue and pink images of electric molecules injected themselves between the mountain and my eyes. He wasn't on LSD, but his mind had learned how to get there, as he later reflected. What if I had not taken LSD ever? Would I have invented PCR? I seriously doubt it. The idea was breathtakingly simple. Heat DNA to separate the strands. Use primers to mark the target region. Add polymerase to copy everything between them. Then repeat. Each cycle would double the copies. 30 cycles would create 1 billion copies of that exact DNA segment. A DNA photocopier. He stopped the car and scribbled the idea on the back of a gas receipt. DNA bombs were exploding in his brain. He could barely sleep that weekend. Monday morning, Mullis burst into the Cetus office. He pitched his DNA photocopier idea. At a company-wide seminar, people started leaving before he even finished. The skepticism was understandable. They'd heard Mullis pitch Kokamami ideas before, and this one seemed too simple. If it worked, surely someone would have tried it already. There had to be a reason this wasn't standard practice. But Mullis pressed forward. In September 1983, he started his first experiment, trying to replicate a 400 base pair fragment of human DNA. Month after month, he couldn't get it to work. Human DNA was too complicated. So he switched to bacterial DNA, just a tiny 25 base pair fragment. For months, he'd separate the strands, attach the primers, add the polymerase, trying to amplify that tiny section. By summer 1984, Mullis thought he had it. Indisputable evidence that his polymerase chain reaction, or PCR, worked. His colleagues weren't convinced. His work was sloppy. No controls, no repetitions. Tom White and another scientist, Norman Arne, looked at his data and concluded that no independent scientists would agree it proved what Mullis claimed. Mullis took the criticism personally. He argued constantly. He even started a fist fight. His credibility evaporated. Everyone wanted him out, but White argued PCR had potential, so the company gave Mullis 1 year to prove it with supervision. White, Arnheim, and another colleague, Angie Ehrlich, assigned other technicians to work with him. This new group worked overtime through the fall and winter. By spring 1985, after months of painstaking trial and error, they cracked it. Definitive proof that PCR was possible. Their sickle cell tests now worked in under 10 hours. But PCR still had a massive problem. Each cycle required heating the DNA to 95 degrees Celsius to separate the strands, then cooling it to 30 degrees so the primers could attach and the polymerase could work its magic, then heating it all back to 95 for the next cycle. The polymerase couldn't survive that heat. It came from E. coli bacteria, which only survive up to 50 degrees. So someone had to stand there and manually add fresh polymerase every single cycle. 30 times per test. It was expensive, time-consuming, and defeated the entire purpose. But a solution had been waiting for 21 years. In 1964, a microbiologist named Tom Brock visited Yellowstone National Park. The vivid yellow and orange colors in the boiling hot springs caught his eye. Could those be bacteria? Everyone said it was impossible. Nothing could live above 60 degrees Celsius. Brock disagreed. He set up a lab next to the springs and brought his undergraduate student, Hudson Fries. 4 days, Fries checked test tubes for signs of life. Nothing. Then on the 4th day, something appeared at the bottom. He shook the tube and saw movement under the microscope. I still get goosebumps, Fries recalled decades later. I thought, my God, I'm the first person in the world that ever sees this. They'd discovered bacteria thriving in boiling water. Brock named it Thermos aquaticus Taq for short. Brock and Fries published their findings and stored a Taq culture in a microbial database, thinking it had no practical applications. 16 years later, in the spring of 1985, Kary Mullis stumbled across their paper. He was searching for a polymerase that could survive PCR's high heat. He'd found it. The Cetus group isolated Taq polymerase and tested it. David G. Gelfand, who purified it, said, "It worked like a charm, better than anything we ever fantasized." The Holy Grail had been achieved. Taq didn't just survive the heat, it thrived in it. And because the temperature never dropped below 50 degrees, the primers only attached to their target sequences. All the background noise disappeared. PCR finally worked. Effortlessly, Cetus realized they had a golden goose, but competitors were catching on. If anyone published about PCR first, Cetus would lose its patent rights. The PCR group urged Mullis to publish a solo paper. It was his idea, after all. But Mullis procrastinated. Afraid of losing credit, the rest of the group published their own PCR research in Science magazine in December 1985. Mullis was listed as the fourth author. He was furious. By the time he finished his own paper, no reputable journal wanted it. "Nature didn't call me," he said. "I wasn't one of the good old boys." Science rejected it too. He accused the group of stealing his work, but they scrambled to give him proper recognition. Mullis became the face of PCR after speaking at a microbiology symposium in 1986. It didn't matter. Shortly after, still angry, Mullis left Cetus. The work continued without him. New PCR machines automate the entire process. Soon, they were in every lab doing DNA work anywhere in the world. PCR revolutionized everything. DNA cloning, vaccines, cancer detection, HIV testing. It saved lives. It supercharged forensics. The tiniest DNA sample could now be amplified. Hundreds of wrongfully convicted people were freed. Thousands of criminals were caught. And right at the center of it all stood Kary Mullis. In 1993, he won both the Japan Prize and the Nobel Prize in Chemistry, but he made PCR his story and his alone. His colleagues called him a fame hog. As Henry Ehrlich remarked, to Kary, rewriting history was more important than writing papers. He seems to have viewed PCR as a means to celebrity. After leaving Cetus, Mullis stopped being a scientist. He used his fame to promote his eccentric views. He talked about glowing raccoons and alien abductions. He wrote a book with a blurb calling him the strangest guy to ever win the Nobel Prize. He even testified as an expert witness in the O.J. Simpson trial. But not all his ideas were harmless. He didn't believe in global warming. He didn't believe in the ozone hole. And he didn't believe HIV caused AIDS. In the early 2000s, South Africa was battling a crippling AIDS epidemic. President Thabo Mbeki fell under the influence of AIDS denialists and invited Nobel Laureate Kary Mullis to weigh in. Under Mbeki's leadership, the government refused to provide adequate treatment. More than 330,000 people died. Kary Mullis passed away in August 2019, agli 74. A year later, his invention, PCR, reached its most significant use yet, as PCR COVID tests helped keep billions of people safe throughout the pandemic. The paradox remains. A terrible scientist who made a world-changing invention. The hidden connection in Mullis's story isn't just about LSD or genius. It's about automation. A machine took his job, freeing him to play with ideas that changed everything. As AI transforms our work, we might be creating the conditions for breakthroughs we can't even imagine. What other Kary Mullises are out there, just waiting for their jobs to disappear? Subscribe for more stories that challenge how we think about discovery. Thanks for watching.